Assalamu alaikum students. I hope all of you will be fine with the best of your health. So, today we are going to learn a new analysis named phylogenetic analysis. In this analysis, I am going to make a phylogenetic tree by using different sequences of protein or DNA. Actually, it depends on the type of your data. If you have a protein sequence or a list of protein sequence, then your phylogenetic tree will be based on this protein sequence list. But in case if your sequences are the DNA sequences, then of course we need to use these sequences. Basically, whenever we do an analysis, we use the data. Therefore, it is called data analysis. And we perform different type of analysis on that data. So today, I am going to perform the phylogenetic analysis on the protein or DNA data. So first thing first, in the first step, I am going to show you how we will collect the data. Either the protein data or the data, DNA sequences or the protein sequences. Actually, it depends which type of sequence you want to take for your analysis. However, there are different ways we can collect the sequences. But in this tutorial, I am going to show you two basic methods. If we take the example of cooking in that case, let's say I want to cook a dish of chicken curry, and if the ingredients, like the main ingredient, I have the chicken, right? If we don't have the main ingredient, then we cannot cook the chicken recipe or any type of chicken curry. So same is the case with that phylogenetic analysis. The main thing is data. Data means the phylogenetic tree basically made up of the sequences. These sequences can be DNA sequences or the protein sequences. We can collect these sequences from the NCBI directly, putting the name of our gene in the NCBI and just copying the FASTA sequences and paste it in a notepad file. Or the other method is simply we can use BLAST. In BLAST, we just simply paste our sequence in the query box of the BLAST, and the resultant sequences are the main sequences which are aligned with the particular sequence or the query particular sequence. So we just need to select these sequences from the results and export them into a new file or a notepad file. So in both ways, we can easily collect the sequences and made a new notepad file containing these corresponding sequences of DNA or protein sequences. So yeah, here you can see I have made two files containing protein sequences. Basically, in first method, I have simply downloaded the sequences from NCBI by just copying and paste. But in the second method, I have used BLAST. So it's just simple. You just need to copy the sequence or the FASTA sequence or the FASTA file and paste it into a new notepad file. So let's use the second method for collecting our protein sequence. In this method, first we will go at the BLAST, and here we have an option to paste the sequence that will be called as the query sequence, or we have another option that we can paste the accession number. So basically it's our choice that how we want to go with that. So let's take our query sequence or the FASTA sequence and paste it into the box of the BLAST. As you can see, BLAST is mostly used for the main purpose basically because it does two things. One is it will align our query sequence with the corresponding related sequences. And the second thing is we can easily download the sequences just by clicking a single option. We will see in the next coming page. Basically, I prefer to use BLAST for this purpose because in that case, we can collect as much sequences as I want. And this is very simple and easy. We don't need to work so hard in order to collect the sequences. So let's see the other options of the BLAST. Basically, in the database corner, as you can see, there is a list of databases from which we can select the sequences. If we want to choose a specific database from which we want that our query sequence should be matched, then we can select the database from this list. Let's see. First, I am taking the protein data bank database in the first option. And let's see the results. As you can see in the results, the similarity is very less and the query coverage is very less. As you can see, the query coverage is just 17%, which is just nothing. And the similarity is, or the percentage identity is very less. So yeah, we cannot go with that, as we want those sequences which have a high similarity and high percentage identity with our query sequences. Well, in that case, our main purpose cannot be fulfilled. So we cannot take these sequences in our phylogenetic tree. So therefore, I am going back and taking another database in order to collect the best sequences for making my phylogenetic tree an excellent picture or a good look. So from here I have selected the non-redundant protein sequences database. In that case, I can think that I will get the maximum identical sequences which will be very identical with my query sequence, because it is a very wide database and it will search from all categories with my query sequence. That's why I like it the most. It's searching and searching, and yeah, here we have got our all results, and these results are surprising. Believe me, these results are surprising. As you can see, the main big difference here in the results. We have a percentage identity of 100%, 98%, 87%, which is a plastic and drastic result, right? So there we have got all the results or the required results which we want to get. And of course, the query coverage is also very good, 100%, 92%, 91%. It is a very good query coverage. It means our whole sequence is being matched and most of the part is similar. So yeah, from here I can select starting 5 to 10 sequences for my phylogenetic tree. Listen, it's very simple and very easy. If we go carefully, we can make it. We can do it, okay? In an excellent way. So from here, we can download these sequences in a new notepad file. I have named this file as a philo2. So this was the main work which we have to do, and we have done this successfully. Congrats! Now let's open the software on which we are going to make this phylogenetic tree. The software is basically MegaX. In that, we will just go first in the file section, or we can also go at the align section, and there we will create a new alignment or build a new alignment, as we know that our sequence is protein sequence. So I have selected here the protein sequence. So from here we have an option in the edit section, and from here we have to choose the option of insert sequence from file. Then we will locate our file which we have downloaded. I have placed it at the desktop. Here I have opened the file just by locating it. So yeah, this is my protein sequence file. These are the colors which will show the differences between the sequences, as these sequences are aligned, but the main alignment we have to do by cluster W. We have an option in the alignment section where we can easily align them all. But first we have to select them. For selecting, we will press Shift button and then just select them all. And from there, we will align them by using the cluster W option. After that, our sequence is aligned. Now we have to export it in the mega format, and we can locate it or place it where we want to place it. See, making a phylogenetic tree is that simple. I am going to put it also in the desktop. Yeah, because at desktop it's easy to locate, right? But in that case you need keep your desktop clean, okay? Okay. I am going to give it the name in mega file so that it could be easy to locate. And of course, a good tip, we need to understand that this file will be in mega format. So when we are going to open it again, we need to find it in mega format. Okay? Just simply save it and close it. After that, I will open it again. When it will be opened, we will see two signs of the files. One is TA and the other is closed data. This will show that our file is successfully converted into mega format and aligned also perfectly. Okay? This is the basically sign that we are going right way. So after that, the last step and the most important and the easiest step, believe me, it's the easiest step, making a phylogenetic tree. I don't know why people think that these things are difficult. If you really have a good interest, you can do that. These are very easy and super easy. So here comes the final look of our phylogenetic tree. We have a lot of options here. As you can see, we can make this tree in our way by using different options, traditional options, circular options. Actually, it depends that in which form I want this tree to be. Even I can change a lot of options. Yeah, like font size, scale length, branch lengths, and we can show up a lot of things in that, like the ambiguous one, the most probable, and a lot of changings we can do according to our own choice. I will try to make it more detailed because the results should be detailed. Yeah, this is a thing which I think that this should be done, because as we see that in our research we have a complete section of results, and in the results we have a lot of things which we need to discuss, which we need to describe, which we need to know. So therefore I am thinking that I will make another part of that uh, analysis in order to make more clear that your results should be different types of data have different types of results, and according to those results we can make different types of phylogenetic trees as well. So basically it actually depends. After that we can simply export it in order to use further. I will tell you that how we can use it further, but for now it's enough. So bye for now.